Namaste children. Today we are going to see about the chapter Fundamental Unit of Life. In this chapter we are going to see many more interesting aspects about cell. Okay. So if you take plants, animals, human beings, microorganisms, all these living organisms are made up of the basic structural unit of life which we called as the cell. If you take cell is called as the basic structural, functional and structural unit of life. Do you know why it is so? Because if you take an entire wall, as I have shown in the picture, it is made up of bricks. So as many bricks join together to form an entire wall, in the same way many cells, they group to form tissues. Many tissues group to form organs. Many organs such as stomach, pancreas, liver, intestine, they group to form an organ system such as digestive system. And many such organ system, that is respiratory system, excretory system, digestive system, reproductive system, they together comprise the entire organism. So that the cells are called as the basic building units. And if you take cells, it was discovered by Robert Hooke in the year 1665. As he was studying the cork cells, he came across many hexagonal shaped units, many small structures which he called it as cell. In Latin language, the word cell refers to little room. So if you take, while you return back to school, we will be doing experiments in onion peel and our cheek epithelial cells. Okay. So if you take onion, if you cut it into two and you just take a thin piece of epidermal cells in the concave side of the onion, and we will just stain it by using saffron solution to color the cells so that it can be viewed. So while you keep it under the microscope, you can see the picture as I have shown in the diagram. So this proves that the plant cells, that is the plant tissues, also possess cells. In the same way, we will be just studying about the animal cells also. We will scrap our inner lining of the cheek cells and place it upon the slide, and we will give stain to it, and we will observe it under the microscope, and we will be viewing a slide as I have shown in the picture. So these two experiments prove that all the living organisms, both animals and plants, are made up of cells. So if you take the body of all the living organisms, they are made up of cells. And cell is a structural and functional unit of life. They can be classified into unicellular and multicellular organisms. See, certain organisms possess only one cell and certain organisms possess many cells. Those organisms which possess only one cell, such as paramecium, amoeba, bacteria and yeast, they all are called as unicellular organisms. The word uni refers to single, one. Whereas if you take certain organisms such as insect, fishes, trees, jellyfish, flowers, plants, and then human beings, they are made up of many millions of cells together. That is why they are called as multicellular organisms. Multi refers to many. So based on the number of cells present in an organism, it can be classified into unicellular and multicellular organisms. Now if you take unicellular and multicellular organisms, they can be differentiated as follows. So unicellular organisms, they are made up of only one cell, whereas multicellular organisms, they are made up of many cells. And all the activities has to be performed, that is digestion, respiration, circulation, transportation, all these things has to be done only by the same cell. Whereas if you take in multicellular organisms, different organs and different tissues will take up the work and they will do it individually. So that the entire work of the organism is being shared in multicellular organism. That is why we say that division of labor is seen in multicellular organism. Whereas if you take in unicellular organisms, division of labor is not seen, because the same cell has to perform all the activities. And if you take in unicellular organisms, the reproduction will occur in the same cell. Whereas in multicellular organisms, a special type of cells which we called as germ cells. Germ cells are nothing but the gametes. Last year you would have studied about male gamete and female gamete, sperm and the ovum, isn't it? So that is what we come to call as germ cells. These germ cells, they take part in reproduction. And under unicellular organisms, you can even give certain examples such as amoeba, paramecium and euglena. Whereas multicellular organisms comprise worms, insects, birds, human beings, etc. So, as I said, cell was discovered by Robert Hooke in the year 1665. It resembled the structure of honeycomb. So, in the first slide I showed you the honeycomb structure, that is hexagonal shaped structure, isn't it? And following Robert Hooke, many scientists have started to discover many things related to cell. So, in the picture I have shown you the bark cell. So, while he was studying about the bark cells only, he came across the cells, structure of cells. So, if you take the other related discoveries, that is Anton von Leeuwenhoek in the year 1674, he discovered bacteria. And Robert Brown in the year 1831, he discovered nucleus. Purkinje in 1839, he discovered protoplasm. Skleddin and Schwann, two persons, they proposed the cell theory. That is Skleddin in 1838 and Schwann in 1839, they proposed cell theory. Cell theory states that all the plants and animals are composed of cells. Whereas in the year 1855, Virchow expanded the cell theory, stating that all cells arise from pre-existing cells. And if you take cells, they are very minute and they cannot be seen with our naked eyes. So, we need an instrument to view those cells, to study about those cells. So, we use microscope. So, microscopes are high resolution instruments which are used to observe very fine minute specimens. Okay, and if you take microscopes, they are of two kinds, that is light or compound microscope and electron microscope. If you take light or compound microscope, which we use in our laboratory, okay, you would have studied many things under light or compound microscope in our school, isn't it? So, that one we call it as light microscope. Here, that is many lenses are combined together and its magnification power, it ranges from 300 to 1500 times. It uses light to illuminate the object. And if you take light microscope, there are different parts present in it, that is the eyepiece through which we see. Okay. And this is an objective lens, that is it is nearer to the specimen, that is the object. So, it is called as objective, and there is an arm to carry the microscope from one place to another, and this is the base of the microscope. Near to the base you can see a small mirror. So, upon the mirror only, the mirror will be adjusted so that the light rays will be made to be exposed towards the light rays. The mirror will be adjusted in such a way so that the light rays are incident over the mirror. So, the incident light rays will be reflected back towards the stage upon which the specimen is placed. Understood? And through this, through the eyepiece, the specimen will be observed. So, these are the different parts of a light or compound microscope, and there are two adjustments. One is fine adjustment, another one is coarse adjustment. So, if you take the magnification power of light microscope is about 300 to 5500 times. So, resolution capacity or magnification power we used to tell. What it is actually, how many times it can be made bigger. Okay. So, that is what 300 to 1500 times. If you take electron microscope, when compared with light microscope, this electron microscope is of bigger in size, and these used to study the even more finer details about the cell, inside the cell. Okay, the cellular structures will be observed. Okay, so very minute details can be viewed under electron microscope, and its magnification power is about 1 lakh times to 5 lakh times. You just imagine how much resolution capacity it possesses. Okay, and this uses electrons for illumination. As the light microscope uses light source for illumination, electron microscope uses electrons for illumination. And if you take kinds of cells, there are two kinds of cells, that is prokaryotic cells and eukaryotic cells. So always you remember the word pro refers to primitive. Okay, karyon refers to nucleus. So the cells which possess a primitive type of nucleus, we call it as prokaryotic cells. So how the primitive nucleus will be seen? So if you take primitive kind of a nucleus, it won't have nuclear membrane, it won't have nucleolus, and the cell do not possess membrane bound organelles in it. So these kinds of features are present in prokaryotic cells, and the size of prokaryotic cells will range from 1 to 10 micrometers. See here I have given micrometer, and nucleoid is present. So nucleoid means undefined nuclear structure, we call it as nucleoid. Okay, if you take a well defined nucleus, means it will be having nucleoplasm, nucleolus will be present, and nuclear membrane will be present around it, isn't it? Whereas if you take nucleoid, they won't have, see the diagram I have shown, no nuclear membrane at all. Just we have to consider that this central region is nucleus, isn't it? Yes. So that nucleolus is absent, nuclear membrane is also absent here, and membrane bound organelles such as mitochondria, plastids are also absent, isn't it? Yes. So this one we call it as prokaryotic cells. Membrane bound cell organelles are absent, cell division by fission or budding, and they lack nuclear membrane, example bacteria. Is it clear? In the same way, eukaryotic cells, U means true, karyon means nucleus. So the nucleus which have a true nucleus, that is the cells which have a true nucleus, we call it as eukaryotic cells. Generally the size will vary from 5 to 100 micrometer, and well defined nucleus is present, and here more than one chromosome is seen, nucleolus is present, nuclear membrane is present, and many membrane bound organelles. See here Golgi apparatus, and then that is endoplasmic reticulum, okay, mitochondria, plastids, everything will be seen here. So because this is called as eukaryotic cells, understood? So example plant cell and animal cell. So if you take division of labor, labor means work. So the work of the entire organism will be divided among different parts of the organism. For example, if you take stomach helps us to digest the food, and if you take heart helps us to pump blood to all parts of the body, and if you take lungs help us to respire, and if you take brain, it controls all the activities of our body. So there is a particular work for each and every specific organ. So all these organs work together so that the entire organism can make its function properly, isn't it? So this is what we call it as division of labor. So if you take multicellular organisms, this division of labor will be seen. Whereas in unicellular organism, the same cell has to perform all the functions of digestion, respiration, circulation, reproduction, everything it has to be carried on its own. So division of labor is not seen. And we can tell that we can see the work being shared upon by different cell organelles such as ribosomes, endoplasmic reticulum, okay, lysosomes. So many organelles are there within a cell. So they will be doing protein synthesis, or one packaging the food, okay, so and transporting the food molecules. So like this, various activities are performed between different cell organelles in a unicellular organisms. And if you take the cell shape and size, always remember, in our body various kinds of cells are present which belongs to different shape. If you take, I have shown here many cells, that is sperm cell, egg cell, nerve cell, then muscle cell, and then fat cells, and then blood cells, okay. So mesophyll cells, so many kinds of cells, columnar cells, many cells are I have given here, isn't it? If you take each one, they differ in their shape and size. If you take sperm cell, it is elongated and it is thin and it has a pointed end. So its main function is to swim about to reach the egg so that it will fertilize the egg. And if you take, that is why it is slender and long with a tail. In the same way, if you take an egg cell, it is globular in shape and it has to get converted into an embryo after getting fertilized by the sperm cell. That is why it is in globular shape. In the same way, the nerve cell, you can see the nerve cell to be branched, isn't it? So why it is branched? Because only if the nerve cell possesses branches, the nerve impulses can be easily be transported from, that is transmitted from one part of the body to another part. Understood? And if you take the blood cells are disc shaped. So they help us to flow into the blood vessels. Okay, that is why it is of disc shape. So that there is a the shape of the cell is entirely dependent over the function it performs. Shape and size of the cell depends on the function they perform. The cells vary in their shapes such as spherical, polyhedral, spindle, elongated, branched or discoidal. The cell sizes vary from small bacterial cells 0.2 micrometer to 5 micrometer. 1 micrometer is equal to 1,000th of a millimeter. Is it clear for you? Then if you take cell, so if you take any typical cell, there are basically three major functional regions. They are cell membrane or plasma membrane, and in addition to the plasma membrane, cell wall will be present in plants. All of you know that. We have learnt in 8th standard, isn't it? And second functional region is nucleus and the other one is a cytoplasm. Cytoplasm is a jelly-like matrix in which we can see living structures of cellular components, and then inclusion, cell inclusions, that is a non-living component we have seen to be present in cytoplasm, isn't it? And nucleus is the controlling center of the entire cell. So these are the three major functional regions present in a cell. So first one, if you take plasma membrane, they are made up of lipids and proteins. Okay, actually it is called as the phospholipid bilayer. So it is seen as a two layers together. See, bilayer, one layer here, another layer here. Okay. So if you take the plasma membrane units, you can see one head and two tail structures will be present. And it is otherwise called as the selectively permeable membrane, because it selects certain molecules only and it allows it to enter into the cell. Okay. So whatever substances that enter the cell, such as oxygen or carbon dioxide or water, it will be entering only through plasma membrane. Understood? So it is called as the living, thin, delicate, elastic membrane, and it is called as the selectively permeable membrane. It is made up of phospholipids, okay, proteins, cholesterol, polysaccharides. So mainly you just have in your mind, plasma membrane is made up of proteins and lipids together, and it permits the entry and exit of only some materials in the cells, and it play an important role in biological activities such as osmosis, diffusion and endocytosis. So osmosis, so we will be studying about these three processes now. So if you take endocytosis, you would have learnt about nutrition in amoeba, isn't it? How amoeba gets its food. So how amoeba gets its food? By using, if it wants to engulf the food nearby, it will just put its plasma membrane into two pseudopodia and just engulf the food, and a food vacuole will be formed within the amoeba, isn't it? So this process only we call it as endocytosis. Endo means inside, cytosis means cell. So inside the cell the food will be engulfed. So this process only is called as endocytosis. So here also plasma membrane play an important role, isn't it? So next slide we are going to see about the osmosis and diffusion. If you take diffusion, I will explain it with a small experiment. You just take a beaker of water and you just add any colored ink, red or blue or pink, any color you can add. You can within few seconds you can see that coloration ink molecules will spread up quickly throughout the water in the beaker, and it will be moving until it gives a uniform coloration throughout the water in the beaker, isn't it? So it do not stay in one place. The ink molecules will immediately spread to all parts of the water so that the concentration coloration will be the same throughout the water in the beaker. So what happens? The ink molecules from the that is higher concentration, it moves towards the place where it is low concentration. Okay, so the molecules of ink, it moves from higher concentration towards lower concentration in order to make the concentration to be uniform. Understood? So this process only we call it as diffusion. See here, the spontaneous movement of molecules from a region of higher concentration to a region of lower concentration leading to uniform concentration is called as diffusion. And this diffusion takes place in all the phases such as solid phase, liquid phase and gaseous phase. Gaseous exchange in unicellular organisms also takes place through diffusion. And if you take the next process, that is osmosis. So in this experiment I have shown here one semi-permeable membrane is placed in between two solutions. So here only water is present. This side of the semi-permeable membrane, that is sugar solution is present. That is sugar mixed with water. These white molecules are sugar, black molecules are water molecules. Okay, so if you just see in the first diagram, more amount of water being present on this side. So from this side the water molecules moves through the semi-permeable membrane into this side, because it is of higher concentration on that side. So from higher concentration it makes a movement through the semi-permeable membrane towards the region where the water concentration is less. Understood? This movement of water from higher concentration to lower concentration through the semi-permeable membrane is called as osmosis. Understood, children? All of you? Okay, so in this way only the water also enters from the soil into the roots through the root hairs. Okay, so osmosis plays an important role in absorption of water by the roots from the soil. And if you take types of solution, that is there are three kinds of solution, that is hypotonic solution, hypertonic solution and isotonic solution. So if you take hypotonic solution, that is it is a very dilute solution. So more amount of water will be there and less amount of solute will be there. So if I am going to take a water, that is a sugar solution, sugar is the solute and water is the solvent. Okay, if I am going to add just 1 spoon of sugar in a very big beaker of water, means it is a very dilute solution of sugar solution. That one we can call it as hypotonic solution. Hypo means very less. Okay, tonic means concentration. In the same way, if I am going to take a glass of water and I am going to take entire packet of sugar to be dissolved in that entire glass of water, means what will happen? The sugar molecules will be more and water will be very less, isn't it? It becomes a concentrated solution. So this solution we come to call as hypertonic solution. Okay, so can you able to understand? So isotonic solution means what? So if the concentration of water will be same. Okay, so this one I will explain to you by placing you just take any animal cell or plant cell and you just place it over the hypotonic solution. So what will happen to the cell if it is placed in hypotonic solution? Hypotonic solution, it contains more water. So the amount of water which is present outside the cell is more when you compare it with the water concentration inside the cell. So water has to move from higher concentration towards lower concentration. So from the external medium, the water will be entering the cell, as a result of which the cell starts to swell up. Understood? Is it clear for you, children? Yes. In the same way, if the cell is going to be placed in hypertonic solution, what will happen? The amount of water which is present inside the cell is more when you compare the amount of water outside the cell. So that water from the cell will make its movement towards the external medium. So that as a result of which the cell will shrink. Understood, children, this one? So if the cell is placed in hypertonic solution, it swells up. If a cell is placed in hypertonic solution, it shrinks. So now let us see what happens if if a cell is going to be placed in isotonic solution. So in this isotonic solution, what happens? The amount of water present inside the cell will be as the same as the amount of water which is present in the solution. Then what will happen? Water movement takes place between the cell and the medium, but the amount of water molecules escapes outside the cell will be equal to the amount of water molecules which enter the cell. So there won't be any change in the cell. The cell remains the same. It do not get swollen, it do not get shrunken. Understood? Yes? See, this you can try by using dry grapes at your home. So you just you take 3 to 4 dry grapes and you just soak in water for overnight. Very next day morning you just see it, you can observe that the dry grapes have swollen. Isn't it? So why this happens? Because the water from the external medium, it enters into the dry grapes. This makes it to get swollen. Okay. In the same way, if it is going to be placed in strong sugar syrup, what will happen? The dry grapes will shrink because water from inside the dry grapes will enter into the external medium. So it results in shrinking up of dry grapes. Understood, children? Then if you take cell wall is the external membrane which is present in plant cells, and cell wall is made up of a complex substance which we called as cellulose. This cellulose provides structural strength to plants. Okay. So if you take cell wall, that is the main function of cell wall is to maintain the turgidity of the cell. That is, it helps to withstand very high water pressure. When water enters the cells means the cells will bulge, isn't it? So it protects the cell without bursting. Understood? So the cell swells and build up pressure against the cell wall. The wall will exert an equal pressure against the swollen cell. So in this way only the cell is getting protected without getting burst out. Okay. And if you take cell wall, I want to tell you one important phenomenon regarding cell wall. So whenever a plant cell is placed in a concentrated solution, that is hypertonic solution, means what will happen? The water from the cell will make its movement towards the external medium. So as a result of which the cell will shrink. Isn't it? So during the shrinking up of the cell contents, what happens? The nucleus and the cytoplasm will come towards the center of the cell. It will come away from the cell wall. So this phenomenon we come to call as plasmolysis. So if you take cell wall, plant cells possess a rigid cell wall outside plasma membrane, and cell wall is non-living and permeable. It determines the shape of the cell. It is made up of fibrous polysaccharide called cellulose. The functions of cell wall are it makes the plant cells turgid, it provides mechanical strength for support, and it is permeable to water. So if you take plasmolysis, when a living plant cell loses water through osmosis, there is a shrinkage of the protoplasm away from the cell wall. What is protoplasm? The nucleus along with cytoplasm together we come to call as protoplasm. So here in the diagram you can able to see that the protoplasm, it comes away from the cell wall, isn't it? And it is concentrated towards the center. That is why this process is called as a plasmolysis. It comes out, it comes away from the cell wall. So in the key, this is the keyword, it comes away from the cell wall, that is a keyword. Okay, yes. So if you take the cell, we have seen here about what is cell, who discovered cell, and many discoveries related to cell, and we have seen about plasma membrane, its nature, and the process of diffusion, osmosis and endocytosis we have seen, and the three kinds of solutions, hypertonic, hypotonic and isotonic we have seen. And if you take this one, that is a cell wall, we have even seen about the plasmolysis. And now we can see about the certain worksheets for practice. So these worksheets you can work out. Okay, and you can easily find the answers only if you go through the lessons. It can be easy for you children. So these kinds of questions you can expect. Okay, so once if you do all these questions, if you attend all these questions, it will become easier for you. So the entire lesson can be learnt very well. Okay, yes. Thank you children. We will see in the next session.